Before I explain the process of Notch delta signaling, it is important to understand the steps in cell differentiation. The process begins with the sensory organ precursor, or SOP cell. The SOP cell has a high concentration of NUM, the protein responsible for cell differentiation. Prior to SOP cell mitosis, the NUM protein accumulates asymmetrically on one side of the cell. The two derived cells are the 2A cell and the 2B cell. The 2B cell inherits most of the NUM protein, while the 2A cell inherits very little NUM. The 2B cell then undergoes the same process as the SOP cell, where NUM is segregated to one side of the cell prior to cell division. The 2B cell divides into a sheath cell and neuron cell, where most of the NUM is localized. The 2A cell divides into the socket cell and the hair cell. The Notch delta signaling pathway begins with two cells, one having Notch, a transmembrane receptor, and the other having delta, a transmembrane ligand. Cells begin with about the same amount of Notch and delta, but one cell will have a slightly higher concentration of NUM, leading to the suppression of Notch and the expression of delta. This can happen due to either an intrinsic biasing signal, which, as shown on the slide, has a higher concentration of NUM within the cell, or an extrinsic biasing signal, which occurs due to a higher concentration of NUM around one cell. Notch delta signaling begins with the delta ligand binding to the Notch receptor, causing an enzyme called an extracellular protease to cleave the extracellular portion of Notch. Once the extracellular portion of Notch is cleaved, an intracellular enzyme called gamma secretase will cleave the intracellular domain of Notch. The intracellular domain of Notch will then move to the nucleus, where it binds to its nuclear partners, mastermind and repressor of hairless. It will then express target genes, which upregulate Notch and downregulate delta. This causes a feedback loop, where cells with high concentrations of NUM have downregulated Notch and upregulated delta. These cells then cause an upregulation of Notch in nearby cells, which upregulate Notch themselves as well, leading to downregulation of delta. Cells that exhibit low Notch signaling will then become neurons in the Drosophilus sensory organs, and cells with high Notch signaling will become sheath cells. Finally, different mutations of NUM or overexpression of NUM can affect the differentiation of cells. In NUM protein mutants, the SOP cell will divide into 2A cells. These cells can then either differentiate into socket and hair cells or strictly socket cells. Likewise, SOP cells that exhibit overexpression of NUM will divide into 2B cells, which will then differentiate into sheath and neuron cells.